Okay, so today I'm so elated to have our guest lecturer, Dr. Danik Martirosyan, the president and founder of Functional Food Center, FFC, Dallas, Texas, and Functional Food Institute, San Diego, California, and also the editor of several textbooks and the one that we're using in our class, The Introduction to Functional Food. His education and leadership positions are very impressive. His dissertation was approved by the highest attestation commission at the Council of Ministers of the Soviet Union in Moscow on July 25th, 1990. He was then granted a doctor's degree in biology. From 1991 to 1993, he worked as a postdoctoral research scientist in the Armenian Ministry of Health, Yerevan, Armenia. From 1994 to 1996, he worked at the Kazan State University, Kazan, Russia. Throughout his time as a researcher, he learned vital skills and gained valuable experience in the field of immunology, biochemistry, membraneology, physiology, food science, and health nutrition. He also worked as an extraordinary ability scientist in Dallas, Texas, in the field of functional food science from 1997 to 2007. It was in 1998 that he founded the Functional Food Center, Incorporated, FFC, to better research and teach about functional foods, bioactive compounds, and the development of healthy and functional food products. Since its commencement, the FFC has developed fundamental principles for functional food science. He also served as clinical professor, Department of Food and Nutrition Science, Texas Women's University, Denton, Texas, 2005 to 2010. In 2010 to 2014 as an assistant professor in the Department of Internal Medicine in University of Texas Southwestern Medical School, Dallas, Texas, working with two Nobel Prize winners, Michael Brown and Joseph Goldstein. As founding and managing director in 2005, he founded the Food Science Publisher and his first set of books on functional food, bioactive compounds, and chronic disease, published under his direction, which has published more than 40 books, including 9 textbooks, 3 academic journals, more than 70 scientific articles, 20 semi-annuals, and 122 brochures on functional food science. He has created the peer-reviewed open access journal, Functional Food in Health and Disease, in 2011. His second journal, Bioactive Compounds in Health and Disease, was 2018, and then the Functional Food Science peer-reviewed journal. Since 1991, he has been working on practical and theoretical aspects of functional foods, bioactive compounds, and functional food science, being one of the founders of functional food science. He has continually initiated discussions in a variety of publications about the necessity of and the definition of bioactive compounds and functional foods regarding functional food science as a separate branch of science. Therefore, the author of the definition of functional food. In 2004, he and his team organized the first international conference of the Functional Food Center as part of the series on functional foods and chronic disease. From then on, he has organized and chaired 29 international conferences jointly with the best universities in the United States: Harvard Teaching School, Harvard Medical School, UCLA Los Angeles, UC Santa Barbara, USDA, ARC, USA at the University of San Diego, California, and also in Japan, Germany, China, and with the USDA.
Introduction, summary, and goal of presentation. Second point going to be bioactive compounds, and we're going to speak in more about description of bioactive compounds and discussion of dosage and timing also. So then we will speak about quantum theory, application of quantum theory in food science. Then we will move to Tempus theory, which is actually about timing, how long should take consumption of functional food product. And finally, we will do our conclusions. So with introduction, I would like to say that with the increase of chronic diseases, functional food products have shown to become part of the solution, because as we know, medication and consumption of drugs not always is resolving situation, and also we have in a most of the time we have in a side effect. So certainly, especially for when we consume for a longer time, there is a bigger chance to get side effect and also having some other problems. So that was the actual one of the biggest reasons that in Japan, starting 1980s, started this science, functional food and related science, because people becoming older and then they live a longer life, but with many diseases, and so it is logical to consume some food and food product which could be helpful in managing somehow disease rather than consuming drugs. So here our goal going to be advance the classification and regulation of functional foods, the importance of bioactive compound dosage in functional food product, establishment of appropriate dosage of bioactive compounds, and using quantum theory of food science, and then exhibit how time can be influenced the effectiveness of bioactive compound using the Tempus theory of functional food science. So very shortly about ingredients of functional foods in United States, in US dollars. So we don't want to focus much on this, but only one slide. So I would like to say that marketing people usually saying that there's over $100 billion sales. But of course, as a professional, we are not sure yet, since we do not have a real definition of functional foods. So the only thing we know that functional food regulated in Japan, and each year in Japan, they are consuming functional food up to $7-8 billion. And it is difficult to say how about in other countries, including United States, because we do not have a special category of food. So it is difficult to say. So this graph provided by some companies who mostly do marketing rather than they creating functional food. So for that reason, we will go to the steps how should be a real created a real functional food product, and how we're going to deal with them, how we're going to know if they are, if ideally we have functional food product in our hands before we're going to calculate how much we sell in each year and in each country. So here's the definition of functional foods, which we come up at Functional Food Center. So functional foods are natural or processed foods that contain biologically active compounds, which in defined effective non-toxic amounts provide a clinically proven and documented health benefit, utilizing specific biomarkers to promote optimal health and reduce the risk of chronic viral diseases and manage their symptoms. I would like to say that many years we were simply saying that reduce the risk of even not reduce the risk, we are saying manage chronic diseases, but later on we changed because to be honest with you, it is very difficult to manage whole disease when we have severe chronic diseases, like someone have 10 years diabetes or 10 years or several years cancer, and then providing one functional food product, certainly it's not going to be easy to manage disease, but we can certainly manage certain symptoms. So maybe for each symptoms, we have to create one functional food product. So it's not going to be easy create one functional food product which is going to deal for several or for all symptoms. So one other thing is that here we could reduce the risk of chronic diseases, because within the years we understand that FDA did not like when we say in a management or a treatment of disease by functional food. And we understand when even if we can prove that really functional food product can treat or prevent certain diseases, but at the end, according to the FDA, that food gonna become a drug. So for example, if we prove that there's enough lycopene in tomatoes or in rosehip, and in certain condition, those product made from those fruits could manage disease. So still FDA gonna consider that that product become a drug. Actually, this is what we get from the communication with FDA, and I would say that FDA, NIH, and USDA representatives lots of time coming to our conferences, and they also are making lectures. So we have pretty good communication with them, and we understand that, and not making a problem with them. And also this is for our understanding also that it is more likely that in the case we creating a excellent ideal functional food product, so they're going to reduce the risk of chronic disease. At the end, we can say that way, rather than saying that they're going to fully prevent or fully treat chronic diseases. So what we have to do, we have to standardize how to create functional food, not only having a standard definition of functional food in this country or in general, but also we have to standardize steps on how to create ideal functional food product. So we come up with 16-step plan how to create a functional food product. Actually, then we're going to see that we added one more step. So now we're going to have 17 steps because we added one other step related to the time, determining time of consumption. So in generally, we can split on 5 major bigger steps. So you can see those steps. So first it is identify bioactive compound. I mean, we can say group of steps. So then preclinical study of determine an appropriate dosage of bioactive compound and mechanism of action. So then provide clinical trials for dosage, efficacy, and safety. Then providing randomized epidemiological studies. Of course, there's lots of other kind of epidemiological studies which are going to be useful, but ideally, best epidemiological study for functional food science is going to be a randomized clinical trials, and then providing an aftermarket study. So now we can go through quickly. We don't have enough time today going through all these steps, but we can quickly, at least, so you're going to have some idea what is step, those steps are about, and each step what is most important stuff. So, but you can see that now So we come up with 17 steps. So first step, it is going to be, look like someone, okay, let's admit. So first step, so it's going to be establishing a goal of functional food product. Because yeah, we have to start from there. Well, what is the goal? What kind of product we would like to create for what kind of disease, or maybe not for disease, for general health, we would like to create a problem. So the initial step is establishing the purpose of the potential functional food product. Then past studies in the different academic fields are called upon that may suggest promising data. So always we have to see if there is any information, any evidence. So not starting from the scratch, but simply looking if previously was used some product, fruit, vegetable, whatever, which is mentions that they have a beneficial effect. So, and then specificity is promote the goal to be clear and concise. So second step to create functional food product is determines relevant bioactive compound. Yeah, this is one of the most important step. So we have to figure out which bioactive compound is important for particular functional food product. Of course, I have to mention that sometimes we having not one bioactive compound, but several, but at least we have to know about that and which one is a major bioactive compound in that product. So here we have to first tell what are the bioactive compounds, and they are primary and secondary metabolites of natural ingredients, nutritive or non-nutritive that promote health maintenance. So they can be in different amount in different foods and in different functional food products, and many analytical chromatographic and molecular approaches are used to fingerprint bioactive compound. Yeah, certainly. So there's a lot of method to analyze and to verify bioactive compounds. So then the third step is establish the appropriate dosage of bioactive compounds. So dosage defines the therapeutic range that a compound may exhibit positive effect. For example, if you have bioactive compound, vitamin C, let's say, very known. So if a regular range is, daily value is from 70 milligram per day to 100, for women about 70, 80, and for men 100. So starting at a certain point like 200, 300 milligram, so it's starting vitamin C having a therapeutic effect. Biological activity has commonly been linked with a positive effect, but negative effects are also a form of bioactivity. So we can imagine if we're going to take too much bioactive compound, so we may have a negative effect. For that reason, we always have to know what is the over limit for that particular bioactive compound. So if we're going to speak particular for the same vitamin C, so daily value of vitamin C should not be more than 2000 milligram per day or 2 gram per day. And I would like to say that nowadays there is some researchers who are using more than 2000 per day, and even they may have some positive effect, but this is should be considered, with FDA if they really can, and USDA actually, because USDA is the one who put in those investigating and having those over limit situation, what has a dosage daily value per day. So always we have to deal with them. So we cannot simply having a 10 gram of, meaning 10,000 milligram of vitamin C and trying to get some beneficial effect. Of course, sometime when, for example, a person have a severe cancer, I know there's one clinic, at least in United States, who using 20,000 milligram per day, and they're trying to manage cancer. But this is, like I said, this is another situation, so it should be considered with authorities, because it may have, and more likely they're going to have some side effect. So defining functional food dosage is important with the bioactive compound process safety perspective. So later on, we're going to come back to the dosage in more detail. So now, so a little bit about fourth step, which is establishment of appropriate time of consumption of bioactive compound. So we're using a tempus, like a Latin word, which meaning time. So kind of how long time we have to consume any bioactive compound or a ingredient, or at the end finalized functional food product. Because we can't simply use functional food as much as we would like, even it's not drug, but still, since it having a bioactive compounds, so and we have to come up with exact dosage and timing, how long we have to consume that bioactive compound, so we don't going to have a negative effects. So yes, so here what we have that as seen in our squalene experiment, the appropriate dosage and time of consumption for bioactive compound will be a range of dose and time. So depending what dosage we're using, and accordingly we can use less or more time. So we're going to speak a little bit later more. And then when consuming bioactive compounds, the claimed health effect does not occur immediately, but they occur after a certain amount of time. This is meaning that, for example, the consuming squalene, which is another very known bioactive compound, known more than 100 years. So how much, whatever dosage we're going to use, but it's not going to be right away visible, the beneficial effect. So in our experiment we use 200 milligrams, 400, 600, 800 milligrams, and we have different results. But anyways, first day until mostly 2 weeks, we did not see much effect, but within a time, so we have an accumulation, and also we can see after several time, we can see effect of bioactive compounds. So let's move to the next one. So next step is determines the specific pathways and mechanism of action. So always we have to come up with the mechanism of action of bioactive compound, and this is one of the steps that Functional Food Center asking for anyone who working with functional food. So they have to clarify at the end what has the mechanism of action of bioactive compound, and this is same with the regulation in Japan, and that's why we can see any article when they submitting to finalize functional food product. So we can see in published journal, in our journals, that mostly Japanese scientists, so they coming up with molecular mechanisms. So before, because if you did not know molecular mechanism of action of bioactive compound, so it's not going to be considered, it's not going to be approved as that kind of functional food product. So that's one of the most important thing you have to know. So you have to figure out that and document. And if should a negative interaction be observed, then this may cause a ceasing the development. So that's in regulation, and certainly we will bring this for United States also in the case FDA accept our suggestion for functional foods having as a separate category. So then sixth step is establishment of relevant biomarker. So why it's very important, because without biomarkers, how we gonna know if bioactive compounds works or not? So for example, you have in some agent, let's say amaranthine, that we are working for a long time, which could somehow could moderate or manage cholesterol level, but how are we gonna know? So to have that, to clarify that, we have to have certain biomarkers, and those biomarkers, in the case of cholesterol, in the case of, so they're going to be different. Different HDL, LDL, and other kind of cholesterol that we have to measure and say if it is effective. Same as we would like to, yes, see hypertension. So again, this for hypertension could be then those different kind of cholesterols can be biomarkers. So anything you're measuring, you would like to come up with functional food product. So you have to, or finding out if your bioactive compound is good for many certain condition. So you have to first find appropriate and sensitive biomarkers. Okay, let's move to the next. So next step number 7 is choosing the appropriate food vehicle for bioactive compound. So yeah, this is one of the very important and interesting point. So because let's say we come up with, we understood what bioactive compound we have to use, but then how are we going to use that bioactive compound? Where we have to put? So that's meaning we have to find out or any appropriate food vehicle. For example, if you would like to this put in some new formulation, if you would like to put in whatever daily you're consuming, soup, bread, pizza, or whatever. So that's meaning food vehicle. So for each bioactive compound, we find out, so we have to then figure out for which vehicle, food vehicle is work much better. So here is some point, how should be a good food vehicle. So we don't want to stay on this a long time, but you can later on see all these points. So step 8 is provide preclinical studies, studies on efficacy and safety. So certainly, safety issues are extremely important, and especially nowadays, anything you do. So FDA going to be asking you special experimental data. So you have to prove that any product you creating, so they are safe, and mostly those experiments, so we have to do on animal models. So then number 9, step 9, so provide clinical trial for dosage, efficacy, and safety. So as we figure out dosage and efficacy in animal models, so then we have to see if that works in the case of human. And for that reason, we have to do human studies, clinical studies. And I would like to say that there is not enough knowledge how to transfer that amount of bioactive compound you figure out that's going to work in animal models. So it is not exact proportional that you're going to simply multiply if you have 100 milligram, or a gram rat or mouse, whatever. So it's not exactly proportional that you're going to multiply and see human, weight. It's not always that. There's some methods. So you have to go through and figure out what's the best way, how is that going to work, and how much you have to take. But at the end, so within clinical trials, one more time, you're going to figure out dosage, best dosage, and also you're going to see efficacy and safety, if there is any safety issues with that bioactive compound or with that it's a functional food product. So then step 10, create a special label that informs the consumer of the most effective way to consume the product. So this label going to be different from regular label, since you are not familiar more likely with that. So I would like to bring like one example from Japanese Foshu food, food for health. So like in this case is Santari Tokucha Jasmine tea. So you can see that in labels they put those important information approved on January 22nd, 2020, which in regular label you don't gonna put. They put it here, active compound, isoquercetin, 110 milligram, and 500 milliliter bottle. So that much of bioactive compound in 500 milliliter bottle. Yeah, and then proof claim. So what was the claim? They come up with the investigation. So then efficacy studies, what they studied, and also safety. So what they already did studied about safety, and they're gonna put how many weeks, whatever time they did, and also they're gonna put a qualified label of category. So this is what we trying to put ABC. So I think this is from our now steps. We're going to see that at the end we would like to come up and classify functional food by ABC according to the level. Okay, finally we were able to move slide. So we have to educate the market. So what is meaning this step? So simply we have to educate market before we will provide that certain functional food product. Because otherwise, if people did not know what is about your functional food, for which reasons they have to take, how long they have to take, so it is not going to be appropriate, or people they will not buy, or they're going to consume not correct way. So for that reason, in different with regular food, so functional food products should be marketed before you're going to bring to the market. So that's another important step. Yeah. So then step 13, send information to credible third parties and or governmental agencies. So at this point, we asking companies submitting application to FDA and functional food center certainly can be helpful on this. Maybe in advance we can look all information before submitting to FDA. So then if it's approved, so at the end it is going to be established functional food product. So this is going to be product C, so category C. Oops. So yes, so we're going to have category C after that, which is meaning that you go through all steps, including clinical study, after marketing, publishing, verifying molecular mechanism. But in this step, you did not done any aftermarket study or epidemiological study. So then in 16, so you're gonna do epidemiological studies, and if you have, and if you've done epidemiological studies, and everything is right, and the product having a positive effect without much side effect, so then you can be approved for the functional food for the level B. And finally, so if you've done aftermarket study and epidemiological study, so then you will have category functional food product for category A. So then, so we can see 3 categories. This is what we thinking that in United States should be 3 category of functional food. So regular one, category C, so improved one, category B, and finally ideal, I would say, functional food for category A. So then let's stay a little bit more with dosage and timing, how to find. So so then we're gonna spend a little bit time on quantum theory and later on Tempus theory. So we already spoke about bioactive compounds, so mostly we finding an appropriate dosage, and certainly it could be a different dose, not one dose for bioactive compound effective. So then we have to figure out what's the dose working best. I think we have some other slide where we're gonna see how to find the best dosage. So here we saying that we have to find a dosage in preclinical and clinical trials. So then here, so we say what is general quantum theory and how to use in functional food science. So if it's general quantum theory provides information about energy at the atomic and subatomic level, and has some other criteria related to the general quantum theory. And here we're saying that quantum theory of functional food science describe the critical amount of bioactive compound within a functional food that cases chain reaction events to lead a health benefit when consumed. So this means that, you know, when you consuming a little bit bioactive compound, certainly it's not going to work. Or a little bit functional food, not just maybe having an ideal functional food. So then you're consuming 1 day, 2 days, 3 days, certainly it's not going to work. So it's coming a time when you have consuming critical amount, and then it is going to be starting kind of chain reaction. So it's going to start some event. Okay, so we published article, Quantum Theory of Functional Food Science, in Functional Food Science Journal in this March. So if you are interested later on, you can go and read that. And yeah, this is simply saying that we provided series of experiment with squalene, bioactive compound, and measured around 30 different parameters for the patient who have diabetes. And we used different dosage, 200 milligram per day of squalene, 400, 600, and control group Also, we had a healthy group. So in total, we had a 150 50 participants with different groups. And also we continued this study 84 days and measured different parameters for 14 days, 28 days, 56 days, and 84 days. So here is how we find out dosage of bioactive compound. So yeah, you can see in different three group, 200 milligram, 400 milligram, and 600 milligram. So you can see that it at some point it started working. So you can see certain, at this point we did not put what the percentage, but certainly in article we have all those percentage. But you can see that it's starting, affecting on proteinuria, particularly. So proteinuria, one of the bad thing for the patient who have a diabetes, simply having a more protein in kidney, which is very, very bad. But then if we continuing, we providing double dosage, 400 milligram per day. So you can see we have a much better effect. And also, as we providing 600 milligram daily, so we can see best effect. But here, one other conclusion should be that we have to continue, actually, maybe in some other time, we have to come up again with this experiment and using a little bit more dosage, because we can see that it is improving. So it is kind of, I remember it was 26 or whatever percent each improvement, but we may continue and see what. Yes, so then using quantum theory of functional food science, so how we can use, so we can use by determining the critical amount of bioactive compound. So ensuring that consumer are eating functional food with enough bioactive compound to produce the desired outcome that claimed by the product, and not only does understanding the critical amount ensures that product is effective, but also that is safe, so that we are not consuming more than needed. Let's move, see if this is moving. Okay, so continuing this, so it could be useful, this theory, for another reason, so that we're consuming enough bioactive compound. I think this is the same one, actually. For some reason, it's not moving. I don't know what the reason. It's moving so slow. So another 2 point, how quantum theory can be useful in functional food science for the following. So determining the critical amount of bioactive compound at which accelerated fission occurs can be established, the dose at which it can become toxic and create adverse effect, therefore negatively impacting health. And then functional foods should contain enough bioactive compound to create chain reaction. So that's, and toxicity, we already spoke about this. So then let's a couple slides about Tempus theory. So simply, quantum theory of functional food science can aid establishing proper dosage of bioactive compound, but quantum theory of functional food science does not speaking about intercount of time. So for that reason, it's very important to find out exact time we need, because like I showed, so it's going to be happening very different thing on 14 days, 28 days, 56 or 84 days. So we have to figure out which days are the best. So time can influence the effectiveness of bioactive compound by the amount of time it takes for it to be effective and how long this effect lasts. Yeah, so this is what we already show in previous slide, but with different time. So now we would like to say, and also here is one of the most important thing. So you can see that first, when we use squalene to manage immunoglobulin A, so affecting on immune system. So we can see that in first 28 days, it wasn't much visible. But then from 28 to 56 days, so you can see effect of squalene of different dosage. But then also we see that after 56 days, so there's not much different. So this is meaning using this Tempus, so we can figure out what are the best time to consume functional food. So there's no reason actually after 56 days consuming, so because you're gonna get best value at 56 days. And yeah, certainly you can see here different dosages. So of course it is best dosage was 600, so then 400. Similar effect, so we can see different days. So you can see increasement up to 56 days, and then there's not much increasement. But in the case of 200, so you can see since amount of squalene is small enough, so it is still increasing. It is still increasing. So from this experiment, so we can see that it is better having a 400 milligram to deal with HDL cholesterol. So no reason to have a much higher dosage. I mean, slightly, very slight different. You can see between 400 and 600. Yeah, but you don't want it. I mean, that's actually medical doctors should be determine this. So another thing is, so we did this experiments, so to see how it is affecting on superoxide dismutase. So you can see that even 84 days experiment wasn't enough at this point using any of those dosage, 200, 400, and 800. So even we can see that 84 days is much better, but we can see that it's increasing. So this is meaning that actually after our experiment should be continuing to figure out. Otherwise, if we do not have this knowledge, this theory, so we're going to simply saying that, yeah, we have excellent, result. We can see that using squalene 84 days, so we can, effect on superoxidized dismutase. But yeah, at this point, we can see that we have to continue experiment, or it is might be better simply having a bigger dosage of squalene and using for the same amount of time. See what's the best time to use this bioactive compound. So this is about squalene, how affecting on cellular energy is very important, very important slide, because one of the biggest deal we have in this with the diabetic people that they don't have enough energy and look like a molecular level. So level of ATP is less, and at this point we are able to show that consuming squalene, so it is affecting on ATP level. And yeah, you can see this is 200 consumption, This is 600 and 400. So I would say anyways, again, up to 56 days. So you can see very good difference. And then it is slow, so very slow increasement. So finalizing, so Tempus theory, functional food center is that along with quantum theory of functional food science, a Tempus theory of functional food science should be used to determine the appropriate time consumption of bioactive compound at which functional food product can be create beneficial effect on one's health and or manage symptoms of disease. I think this is our last slide, and thanks so much for your patience. So here is summary. So finding the appropriate dosage of bioactive compound is an important step in the process of development of functional food product. And second point is that clinical research and quantum science can be used to help determine the dosage that provides the most positive effect. And third, time is an important aspect of determining the effectiveness of bioactive compound and should be implemented. And future studies should consider the quantum theory and Tempus theory of functional food science to gain a better understanding of the critical amount of bioactive compound. And finally, using that, we were able to figure out which parameters can be managed and best time. So for HDL, we figure out that 56 days is best time. To SOD is 84 days we have to use, and for proteinuria also 84 days was the best time to use squalene with the amount of 600. So this is all, and thanks so much for our employees and students who helped me. So